Pharmaceutical companies conduct clinical trial analysis in a highly dynamic environment, and the results have to be processed faster for further process execution. Similarly, governance bodies and regulators, like FDA, conduct separate trials to bring the product in the market after undergoing stringent evaluations. The clinical trials produce machine-generated unstructured content after each lab trial. This content is stored at predefined shared paths or URLs belonging to the FDA, from which it needs to be extracted in a structured format and integrated with downstream systems. This video demonstrates how TruAI uses advanced artificial intelligence techniques, such as NLU, to automate data extraction and integration seamlessly in an agile environment. The screen shows the overall workflow of a typical clinical trial data extraction using TruAI. The analysts receive the machine-generated clinical trial reports as URLs printed in a text file. The analysts feed the input text file that lists the URLs in the TruAI ensemble. The ensemble downloads the PDFs from the respective URLs. The ensemble then separates the required sections of interest for further analysis. The NLU component finally processes the extracted sections to derive meaning and context, such as identifying addresses or making inferences. The ensemble saves the extracted results in JSON and TXT file formats. The JSON file contains structured data, such as detected addresses. The TXT file contains observations, such as negative inferences, etc. Let us see the FDA clinical trial data extraction using TruAI in action. The screen displays the empty folder on the left where the TruAI ensemble downloads the PDFs. The empty folder on the right stores the output files that TruAI generates. The analyst downloads the text file containing URL links to the PDFs from the FTP server. The analyst then executes a code that downloads the PDFs from the URL links into the designated input folder. Subsequently, the analyst executes the main code through a CMD interface. In the background, TruAI processes the PDFs to convert the PDF pages into images. TruAI intuitively detects the sections to be extracted and separates them for further analysis. NLU component then uses advanced techniques to derive meaning and context from the sections. TruAI then saves the results in JSON and TXT file formats. TruAI creates a subfolder for each input file in the output folder. Each subfolder contains the corresponding JSON and TXT files. These files help integrate the extracted data seamlessly with the downstream systems. After the process is completed, the analyst executes a code to move the PDFs and the images of their individual pages into a completed folder. The completed folder displays the images with detected sections, highlighted with bounding boxes, and includes confidence values. The screen now displays one of the TXT files that contains the trial observations. Now, the screen shows one of the PDF pages with bounding boxes and confidence levels with which TruAI extracts the data in TXT and JSON formats. Now, the screen shows the output files. This is the JSON file output extracted from one of the PDF images shown on the right-hand side. TruAI improves the speed and accuracy of the clinical trial data extraction by 80% as compared to manual effort. It automates the seemingly impossible task of extracting structured data from machine-generated unstructured data. To know more, visit Datamatics.com.